welcome students to the Rashmi's classes. Today's we'll discuss on NCERT Class 12 Biology.
NCERT Class 12 Biology. Principles of Inheritance and Variation. 4.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel where he crossed tall and dwarf pea plants to study the inheritance of one gene, figure 4.2. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial 1 progeny or the F1. Mendel observed that all the F1 progeny plants were tall, like one of its parents, none were dwarf, figure 4.3. He made similar observations for the other pairs of traits. He found that the F1 always resembled either one of the parents, and that the trait of the other parent was not seen in them.
Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4th of the F2 plants while 3/4 of the F2 plants were tall. The tall and dwarf traits were identical to their parental type and did not show any blending, that is all the offspring were either tall or dwarf, none were of in-between height, figure 4.3.
Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation while at F2 stage both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down unchanged, from parent to offspring through the gametes, over successive generations. He called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles, i.e., they are slightly different forms of the same gene. If we use alphabetical symbol S for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, T is used for the tall trait and T for the dwarf, and T are alleles of each other. Hence, in plants the pair of alleles for height would be TT, TT, or TT. Mendel also proposed that in a true breeding, tall or dwarf P varietith allelic pair of genes for height are identical or homozygous, TT and TT, respectively. TT and TT are called the genotype of the plant while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype TT? As Mendel found the phenotype of the F1 heterozygote TT to be exactly like the TT parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other, as in the F1, and hence is called the dominant factor while the other factor is recessive. In this case T, for tallness, is dominant over T, for dwarfness, that is recessive. He observed identical behavior for all the other characters/trait pairs that he studied. It is convenient, and logical, to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use T for tall and D for dwarf because you will find it difficult to remember whether T and D are alleles of the same gene/character or not. Alleles can be similar as in the case of homozygotes TT and TT or can be dissimilar as in the case of the heterozygote TT. Since the TT plant is heterozygous for genes controlling gene character, height, it is a monohybrid and the cross between TT and TT is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that, when the tall and dwarf plant produce gametes, by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chance of a gamete containing either allele, as has been verified by the results of the crossings. In these way the gametes of the tall TT plants have the allele T and the gametes of the dwarf TT plants have the allele T. During fertilization the two alleles, T from one parent say, through the pollen, and T from the other parent, then through the egg, are united to produce zygotes that have one T allele and one tall A. In other words, the hybrids have TT. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram called Punnett square as shown in figure 4.4. It was developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written in two sides, usually the top row and left columns. All possible combinations are represented in boxes below in the squares, which generates a square output form. The Punnett square shows the parental tall TT, male, and dwarf TT, female, plants, the gametes produced by them and, the F1 TT progeny. The F1 plants of genotype TT are self-pollinated. The symbols and % are used to denote the female, eggs, and male, pollen, of the F1 generation, respectively. The F1 plant of the genotype TT when self-pollinated, produces gametes of the genotype T and T in equal proportion. When fertilization takes place, the pollen grains of genotype T have a 50% chance to pollinate eggs of the genotype T, as well as of genotype T. Also, pollen grains of genotype T have a 50% chance of pollinating eggs of genotype T, as well as of genotype T. As a result of random fertilization, the resultant zygotes can be of the genotypes TT, TT. From the Punnett square it is easily seen that 1/4 of the random fertilizations lead to TT, 1/2 lead to TT and 1/4 to TT. Though the F1 have a genotype of TT, but the phenotypic character seen is tall. At F2,3/4th of the plants are tall, where some of them are TT while others are TT. Externally it is not possible to distinguish between the plants with the genotypes TT and TT. Hence, within the genotypic pair TT only one character T tall is expressed. Hence the character T or tall is said to dominate over the other allele T or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, though the genotype is TT, and in the F2 3/4 of the plants are tall, though genotypically 1/2 are TT and only 1/4 are TT. This leads to a phenotypic ratio of 3/4 tall 1/4 TT plus 1/2 TT, and 1/4 TT, i.e., a 3:1 ratio, but a genotypic ratio of 1:2:1. The 1/4, 1/2, 1/4 ratio of TT, TT is mathematically condensable to the form of the binomial expression, X and by 2, that has the gametes bearing genes T or T in equal frequency of 1/2. The expression is expanded as given below. 1/2t plus 1/2 t, 2 equals 1/2t plus 1/2t, x, 1/2t plus 1/2t, equals 1/4 TT plus 1/2 TT plus 1/4 TT. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarfs was homozygous, TT. What do you think would have got had he self-pollinated a tall F2 plant? From the preceding paragraphs it is clear that though the genotypic ratios can be calculated using mathematical probability, by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has TT or TT composition, cannot be predicted. Therefore, to determine its genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross on an organism, P plants here, showing a dominant phenotype, and whose genotype is to be determined, is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 4.5 shows the results of typical test cross where violet color flower, W, is dominant over white color flower, W. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get, using the genotypes of this cross? Can you give a general definition for a test cross? Based on his observations on monohybrid crosses, Mendel proposed 2 general rules to consolidate his understanding of inheritance in monohybrid crosses. Today these rules are called the principles or laws of inheritance, the first law or law of dominance and the second law or law of segregation. 4.2.1 Law of Dominance. I. Characters are controlled by discrete units called factors. 2. Factors occur in pairs. 3. In a dissimilar pair of factors one member of the pair dominates, dominant, the other, recessive. The law of dominance is used to explain the expression of only one oft parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 4.2.2 Law of Segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parents contain 2 alleles during gamete formation, the factors, or alleles of a pair segregate from each other such that a gamete receives only one of the 2 factors. Of course, a homozygous parent produces all gametes that are similar while a heterozygous one produces 2 kinds of gametes each having an allele with equal proportion. 4.2.2.1 Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes TF1 had a phenotype that did not resemble either of the 2 parents and was in between the 2. The inheritance of flower color in the dog flower, snapdragon or Antirrhinum sp. is a good example to understand incomplete dominance. In a cross between true-breeding red-flowered, RR, and true-breeding white-flowered plants, RR, the F1, RR, was pink, figure 4.6. When the F1 was self-pollinated, F2 resulted in the following ratio: 1, RR, red, 2, RR, pink, 1, RR, white. Here the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratios had changed from the 3:1 dominant, recessive ratio. What happened was that R was not completely dominant over R, and this made it possible to distinguish RR as pink from RR, red, and RR, white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, i.e., as a pair of alleles. Now, these two alleles need not always be identical, as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on, and in the next chapter, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now here are two copies of this gene, the two allelic forms. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of A substrate S. Theoretically, the modified allele could be responsible for production of the normal/less efficient enzyme, or a non-functional enzyme, or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, i.e., it will produce the same phenotype/trait, i.e., result in the transformation of substrate S. Such equivalent allele pairs are very common. But, if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype/trait will only be dependent on the functioning of the unmodified allele. The unmodified, functioning, allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. 4.2.2.2 Codominance. Till now we were discussing crosses where the F1 resembled either of the two parents, dominance, or was in between, incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by thegony. The gene, I, has three alleles IA, IB, and I. The alleles IA and IB produce a slightly different form of the sugar, while allele I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over I, in other words, when IA and I are present, only IA expresses, because I does not produce any sugar, and when IB and I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence red blood cells have both A and B types of sugars. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible, and therefore, a total of 6 different genotypes of the human ABO blood types. Table 4.2. How many phenotypes are possible? Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, i.e., 3 alleles, governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, B and B. Starch is synthesized effectively by B homozygotes, and therefore, large starch grains are produced. In contrast, BB homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seeds, BB seeds are rund and the BB seeds are wrinkled. Heterozygotes produce round seeds, and so B seems to be the dominant allele. But, the starch grains produced are of intermediate size in BB seeds. So if starch grain size is considered as the phenotype, then from this angle, the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine, in case more than one phenotype is influenced by the same gene.
Thanks for watching the video. Please like, share and subscribe.